All right, welcome back. So where we left off last time, we were talking about plasma membrane and different types of transport into or out of a cell. And the very last topic we discussed was endocytosis and exocytosis. And so those terms are specifically describing the bringing of something into the cell and then the release of something out of the cell with a membrane-bound structure, a vesicle. And now as we move into our membrane-bound organelles, we're going to talk a bit about the endomembrane transport system, which can be a mouthful, but if we break that word apart, endomembrane, so membranes inside, transport system, we're going to be looking at how molecules are moved throughout the cell within these membrane-bound vesicles. So since all of our organelles also have a phospholipid membrane, and our vesicles that are the transport structures moving things throughout the cell have a phospholipid membrane. Those membranes can just fuse into each other or pinch off in the same way that we saw with endocytosis and exocytosis. So if we need to bring something into the cell and take it all the way to the Golgi apparatus and then all the way to the endoplasmic reticulum, we can do so while keeping those molecules contained in their own membrane-bound vesicle without ever needing them to mix with our cytoplasm. And so we're going to start off with talking about this endomembrane system by looking at the endoplasmic reticulum, because most of what is being transported throughout the cell in these membrane-bound vesicles are going to be proteins. And the endoplasmic reticulum does a lot for protein creation and protein modification. So our endoplasmic reticulum is, in most eukaryotic cells, going to be directly connected to the nuclear envelope of our nucleus, so the membrane of the nucleus. So you would find the endoplasmic reticulum close to the nucleus. We're going to see some exceptions to this. There's smooth ER that is free-floating in a lot of our cells. There is some rough ER, endoplasmic reticulum, that we see in neurons specifically, called Nissl bodies. So we'll see that introduced later in Unit 3. But in most of our cells, the endoplasmic reticulum is mainly all part of this interconnected system that is directly attached to the nucleus. And so we have our separation into rough ER and smooth ER, so RER and SER. The rough ER has its name because it has a bunch of embedded ribosomes directly on the membrane of the endoplasmic reticulum. And so the rough ER is going to have ribosomes create proteins, create polypeptide chains, and directly release those into the endoplasmic reticulum, where they will be modified slightly and then packaged into vesicles in the smooth ER, and those vesicles will take them to the Golgi apparatus. Right, so that first part of our endomembrane transport system. So the rough ER is mainly there for the creation of proteins and some modification of proteins. The smooth ER is where you'll see some of our lipid metabolism, so creating new lipids, some of our carbohydrate metabolism, creating structural carbohydrates, and then also creating those vesicles, the vehicles that will carry proteins, carbohydrates, and lipids throughout the rest of the cell. So you can see that why these things got their name, why we have a smooth versus a rough ER when looking at it under a microscope. All those little ribosomes look like bumps on the membrane of the endoplasmic reticulum. And so that gave it a rough looking structure and thus the name rough ER. Whereas the smooth ER didn't have any of those little bumps, so it looks a lot smoother, so smooth ER. I guess just to emphasize, since this wasn't explicitly stated, ribosomes are not only found on the membranes of the rough endoplasmic reticulum. Most of your ribosomes are just free floating in the cytoplasm of your cells, and that is the case for most cells in general. The ribosomes are just free floating in the cytoplasm. They are not embedded onto a membrane like this. So the next stage in our endomembrane system, I guess depending on what molecule we're talking about transporting. So in the case of molecules that have been created in the cell, those are mainly being created in the endoplasmic reticulum, and then they will be carried through vesicles, again, membrane-bound, tiny little vehicles. They will be carried to the Golgi apparatus. And the Golgi apparatus is basically the postal system of our cells. Its main function is protein modification. So getting these proteins into their final tertiary or quaternary structure where they are now going to be functional, mature proteins, as well as directing the movement of molecules. So molecules that are being brought into the cell through endocytosis and molecules that are going to be transported somewhere throughout the cell or leaving the cell through exocytosis, that is all directed by the Golgi apparatus. The Golgi apparatus will modify these vesicles that are carrying the molecules throughout our cell by adding glycoproteins or glycolipids that act as the stamp or the address for where that molecule needs to go. So we can talk about specifics like the cis end and trans end. Where is that? I think that's coming up in these images here. But mainly, I just want you to know the main function here, or the main function of these organelles in general, right? So the rough ER was mainly for synthesizing and modifying proteins. The smooth ER is synthesizing lipids and carbohydrates, and then packaging all those biomolecules, proteins, lipids, and carbohydrates into vesicles. The Golgi apparatus, its main function is to act as that postal system, adding the address, so adding specific carbohydrates onto these vesicles that tell them where to go. It is also responsible for that final modification of proteins. So getting them into their mature form where they are now functional, finalizing them into that tertiary structure, creating disulfide bonds, or their quaternary structure, holding multiple polypeptides together. So yeah, I'm not really going to go through all of the stages here. I guess we can quickly flip through. So we're showing essentially the life cycle of a protein being created and then transported throughout the cell. So all protein synthesis starts with our DNA, our genes, all genetic information, all genetic material are simply instructions for building proteins. So the portion of the DNA that codes for the gene that we're looking at is going to unravel. An RNA transcript, an mRNA transcript is going to be made of those instructions. That mRNA will leave the nucleus through a nuclear pore and will either find a free-floating ribosome or will find a ribosome that is directly embedded into the rough ER. You can see that here. The ribosome will then translate those instructions into a polypeptide, so a chain of amino acids like what you see here. And in the rough ER, that polypeptide chain will start to create its hydrogen bonds into its secondary structure, might start creating there might be other modification into its tertiary structure or the beginning of its tertiary structure within the ER. And then it will be packaged into a vesicle in the smooth ER, and that vesicle will take it to the Golgi apparatus, as we see here. And so on the cis face of the Golgi apparatus, that is the receiving end. And then we have our trans face, which is the distributing end. So these proteins are being brought in to the Golgi apparatus on the cis face and then will leave in their mature form on the trans face. Typically, they will have some carbohydrate attached to them, acting as a signal telling them where to go, such as being embedded into the plasma membrane or being taken to the mitochondria or something. Or they might be added to a lysosome, which will break them apart. So depending on what kind of structure we're moving throughout the cell, the Golgi apparatus can also help with that digestion, that recycling of those molecules by adding them onto lysosomes. They are also responsible for the creation of lysosomes. Right? Many of these proteins may be the digestive enzymes themselves that are going to end up as lysosomes, waiting to destroy our invaders or break down other molecules. And then that vesicle could also go to the plasma membrane and simply exocytose whatever we created, right? So digestive enzymes like lipase and amylase that need to be released into the small intestine to help us digest the food that we've eaten, right? Those enzymes would be created in the rough ER, sent to the Golgi apparatus, and then the Golgi apparatus would direct them to be exocytosed out of the cell. So next, looking at the mitochondria, probably the most well-known organelle within the cell, commonly referred to as the powerhouse of the cell, which is a minimalistic way of looking at what the mitochondria is and what it does. It's a really incredible organelle and very unique in lots of ways to everything else in a eukaryotic cell. we can start with the basics, then I'll go into my tangent about why the mitochondria is so exciting. And so focusing on that powerhouse component, its role as a producer of ATP, okay? A majority of the ATP in a cell is going to come from the mitochondria, because the final two phases of cellular respiration, being the citric acid cycle, aka Krebs cycle, as well as the electron transport chain, both of those occur in the mitochondria. Okay? Without a mitochondria, we could only rely on glycolysis, which makes very little ATP. It's not very efficient. So the mitochondria is great. Okay? It's the most important unique structure of the mitochondria is that it has two membranes. So all membranes are bilayers. So I'm not referring to two layers of phospholipids. I mean, there are two completely separate membranes in the mitochondria. Both of these are phospholipid bilayers. So we have an outer membrane and an inner membrane, which creates extra compartmentalization, extra specialization of the spaces in this organelle. And so all the way within the inner membrane is our matrix, you see referred to here, and that is the location of the citric acid cycle. So this will break down all the rest of the bonds holding carbons together from a carbohydrate or fatty acid, creating or adding electrons, high-powered electrons onto our electron carriers. And then those electron carriers will power the electron transport chain, which happens along our inner membrane with embedded cytochrome proteins on our inner membrane of the mitochondria. And that generates a whole bunch of ATP. So we have special compartmentalization. We have two membranes, so we have multiple spaces in the mitochondria, and you don't see that in any other organelle. So the evolutionary explanation for that is why I'm so excited about mitochondria, and I'll come back to that later. So again, focusing on its role as far as generating cellular energy, or ATP, which we introduced in the last chapter. ATP has three phosphate molecules that have been shoved together. Each phosphate is negatively charged, and so they really don't like being held in close proximity. They naturally want to push away from each other. But using some energy, such as the energy we get from breaking down glucose, we can shove phosphates onto an ADP, and then that ATP can go somewhere else in the cell. It'll easily be broken apart to release that last phosphate group, and that'll generate a whole bunch of energy that can be used to do work. So the major role of the mitochondria is to make a whole bunch of that ATP. And so that all starts with a glucose molecule in our cytoplasm, or in our cytosol, sorry. So cytoplasm is referring to all of the molecules that are in, or the entire internal portion of a cell, really, so including the organelles. The cytoplasm is everything that's in the cell. Cytosol is the liquid, essentially, in between all of the different organelles. So all of the semi-fluid aqueous space that is in between all the organelles in a cell, that is the cytosol. The cytoplasm includes the cytosol and all the organelles. So that's that separation. So anyways, the first stage of cellular respiration, of getting ATP energy from the food that we eat, from the molecules that we consume, is glycolysis. And this always starts with a glucose molecule. So during glycolysis, we're essentially splitting a single glucose in half into two pyruvates. And every time we break a bond holding carbohydrates together, that's going to release the potential energy that was stored in that bond. So when we break this glucose in half, we're going to release a little bit of energy that allows us to make a little bit of ATP. And then some of that energy is going to be picked up by these electron carriers, these coenzymes, that will carry electrons. They're really carrying a full hydrogen, a proton, and an electron, but we call them electron carriers, and they will carry those high-powered electrons all the way into the mitochondria, where they will power the electron transport chain, our last step. So anyways, the main point here is that without the mitochondria, this is all our cells can do to make energy. So we have to utilize a little bit of energy to get this started. So we really only get 2 net ATP from this glycolysis process. And then we still need to recycle our electron carriers, which ends up using more energy later, and recycle this pyruvate, which is going to turn to lactic acid, which is going to utilize more energy in our liver. So glycolysis on its own is not efficient and doesn't make a whole lot of energy. And so being able to utilize the mitochondria or having a mitochondria is going to allow us to make a lot more energy, get a lot more bang for our buck, right? It's a lot more efficient. But we can only go through the next two stages. We can only utilize our mitochondria when our cells have access to oxygen, and thus the importance of those red blood cells, right? Bringing oxygen to our cells so that we can continue to do all the stages of cellular respiration and get the most ATP out of our food that we can. So glycolysis split this glucose molecule in half, generating 2-pyruvate, and we got a little bit of ATP in the process. This pyruvate, both of these pyruvates are then going to come into the mitochondria, again, only if oxygen is present, but they will go into the mitochondria where they will be further broken down through the citric acid cycle. Well, they'll first turn into acetyl-CoA, and then acetyl-CoA will be broken down through the citric acid cycle until all of the carbons that have been bonded together are broken. So we release every individual carbon that was in this glucose molecule. So glucose is a 6-carbon molecule, so we will release 6 carbon dioxide molecules at the end of this process, thus releasing all the potential energy that was stored in those bonds. And most of that potential energy is now being carried by electron carriers. And those electron carriers will power the electron transport chain, which makes a whole bunch of ATP. So let's look at that next phase now. So glycolysis split glucose into pyruvate. Pyruvate comes into the mitochondria. It will immediately lose a carbohydrate or a carbon and be turned into this acetyl coenzyme A. Acetyl coenzyme A will go through the citric acid cycle, also called the Krebs cycle, which will break apart the rest of those bonds. Or there's a lot of chemical reactions happening in this cycle. I don't have the chemistry background and knowledge to fully understand all the different stages and steps, and I'm not asking you to memorize or know all the different stages and steps. The main idea here is that during the citric acid cycle, acetyl-CoA gets fully broken down into carbon dioxide. And as a result of that process, we end up making a whole bunch more electron carriers. So by the end of glycolysis and the citric acid cycle, our first two stages, most of the energy that was in our food that we ate is being carried by these electron carriers. And we've only made 2 ATP. So then our final stage is the electron transport chain. Technically, the final stage is called oxidative phosphorylation. Oxidative referring to the fact that it needs oxygen, and phosphorylation referring to the creation of ATP, so adding a phosphate onto ADP, phosphorylation. And oxidative phosphorylation includes both the electron transport chain and ATP synthase, which are essentially separate processes. But for the purposes of this class and for building an understanding, we can think about all this as the full process of the electron transport chain. And so as the name suggests, the electron transport chain is going to be transporting electrons down a chain of proteins. So like I mentioned before, these NADH and FADH2 molecules that we were creating by breaking or by releasing potential energy stored in those bonds, holding the carbohydrate together, they are carrying these high-powered electrons that will be passed onto this series of proteins embedded in the inner membrane of our mitochondria. So these proteins are going to take that electron and pass it on to the next protein in the chain, and then pass it on to the next protein in the chain. And this is all being driven by these very electronegative oxygens waiting at the end of this chain, creating essentially a magnetic pull that is driving the electrons to move from protein to protein until they eventually are going to end up on those oxygens, and that oxygen becomes water. So oxygen drives this process. Without oxygen present, this can't happen. And so there's no reason to go through the citric acid cycle, so that won't happen, and we can't generate a whole bunch of energy. So oxygen is absolutely necessary for this. It'll pull electrons from one protein to the next. As a result of these electrons jumping from protein to protein along this chain, that is going to cause the opening of a protein channel that will pump protons, this H+, out of the matrix and into the intermembrane space. So every time an electron moves from one protein to the next, a proton is going to move into the intermembrane space. And so by the end of the electron transport chain, we've had a lot of electrons going through this chain, created a bunch of water. And so we've created a very high concentration gradient in the intermembrane space here. We have a whole bunch of protons, a high concentration of protons in this space, and now a low concentration of protons in our matrix. And so that's going to force these protons to want to diffuse back to where they started, get into the matrix. The only way they can do that is through a different protein channel created by a specialized protein called ATP synthase, referring to its role in the synthesis of ATP. Right? This is the protein that is going to be creating a whole bunch of ATP for us. So it has a channel that allows protons to move through it. These protons will simply diffuse through that protein channel. The movement is going to generate kinetic energy that powers the spinning of the second component of ATP synthase. And as that second component spins, it is essentially going to shove a phosphate onto ADP, thus releasing a new ATP molecule. And through all these different steps of this process, one glucose molecule will end up creating or leading to the creation of around 36 ATP just from this part of the process, just from ATP synthase at the end of our electron transport chain. Without this last stage of cellular respiration, each glucose molecule is only making 2 ATP. So when we can utilize our mitochondria and we have oxygen present, we are generating a lot more energy from each glucose molecule. And this is a much more efficient process and thus the powerhouse of the cell. There's just those steps written out for you, the description of ATP synthase written out for you, and a closer look at that protein itself. So like I said, it creates a channel that these protons are going to diffuse through, driven by their concentration gradient. The movement of those protons generates kinetic energy, which is going to spin this rod. And that is going to allow for the shoving of a phosphate onto ADP, creating ATP. And so I remember in my very first Bio 1 course in college that this was like my first indication that I should switch my major. I was a psychology major as my first year as an undergraduate. And when my biology professor was talking about how ATP synthase works, my brain was like, wow, that sounds a lot like an engine or a lot like a wind turbine or a lot like a hydro turbine. And it really is. We just have the movement of some structure which is generating kinetic energy, which causes the movement of a different structure, which leads to the output of electricity, or in this case, ATP. And so it blew my mind at the time that there is this randomly mutated and created thing. Like all of the structures of life are just the result of random mutations or the random combination of molecules in some space. So this randomly mutated occurrence created a structure that functions just like an engine that humans later created at a larger scale before we ever knew how this thing really worked, before we could visualize it. And there's a lot of examples of that, of artificially made technology that functions basically the same way that a microscopic molecule does. And we created these artificial things before we ever really knew about them. So even though our bodies are functioning from these microscopic molecules, this larger human thing created a much larger scale of that same system, of that same structure before we really knew that they existed. And just that whole idea is really cool to me. So before we move on to the nucleus, I want to go back to why the mitochondria is exciting from an evolutionary basis. So besides what I was just saying about ATP synthase, which is exciting, mitochondria are really cool because there is a lot of evidence, or there is strong evidence to support the theory, the idea that mitochondria were originally their own individual prokaryotes. They were their own bacterium-like organisms. And so at some point in early evolutionary history of life, a larger prokaryote basically endocytosed, it engulfed this smaller prokaryote. And rather than destroying it, that prokaryote survived. So it has its own membrane. So the inner membrane of the mitochondria would have been the original plasma membrane of that smaller prokaryote. And it has the second, its outer membrane, which was the plasma membrane of the larger prokaryote that engulfed it. And rather than destroying it, that bacteria survived and created this, oh gosh, why is the term escaping me now? This mutually beneficial symbiotic, there we go, relationship. Right? And so then when that larger prokaryote went to go through the process of mitosis and divide and clone itself, that it was releasing the protein signals that tell the rest of the cell to start replicating. And so that initiated replication for the mitochondria as well, that other bacteria, that other prokaryote that was inside of it. And so the bacteria also replicated itself and ended up on both of those new cells. And so now we have our very first organelle in our very first eukaryotic cell, which was the mitochondria. And I think that's really cool. And so that same idea is theorized to have happened again with a photosynthetic prokaryote, right? And that created what we call the chloroplast. Right? And that went to a different lineage of evolutionary cells, right? The ones that can do photosynthesis, that can capture energy from the sun. But both the chloroplast and the mitochondria have two membranes like this. And that is the strongest supported idea as to why that would be, that they were originally their own bacteria. Other evidence for that is the mitochondria has its own, essentially nucleus, has its own DNA and controls its own replication. Right? So the genes in your nucleus do not regulate or control the mitochondria's function or its replication. It is essentially its own living organism inside all of our cells. So I think that's really cool. Anyways. So now to our last organelle and function that we're talking about in our cells, the nucleus. Okay. The nucleus is often referred to as the brain of the cell. And so just like how in our bodies, our brain is essentially providing the instructions for how the body is supposed to be interacting with the world and taking in information, the nucleus is what's holding all of the genetic material. And that genetic material codes for proteins. And then those proteins control everything else that's happening in the cell. So that's why it's called the brain. So yeah, the nucleus's structure has a lot of similarities with everything else we've been talking about, right? It has a membrane that separates it from the rest of the cell, but it also has some unique structures. So it has its own terms for the cytoplasm of the nucleus called the nucleoplasm. It has a tiny little condensed area inside its nucleus called the nucleolus, which is where we create some ribosomal RNA and generate new ribosomes. It has specialized protein pores, channels called nuclear pores that are very, very selective about what can enter and leave. And then, of course, we have all of our genetic material, our DNA, which has been organized into chromatin, which can be further folded and protected into chromosomes. You can see all that depicted here. So again, that nucleolus is where essentially ribosomes are created. Ribosomal RNA, and ribosomes are just made up of RNA. So ribosomal RNA is created in that nucleolus. The DNA that codes for it is found in that area. And then that RNA can leave and create a ribosome. So the nuclear pore is very specialized compared to other protein channels because we need to be very selective about what is going to interact with our genetic material. We want to reduce the opportunity for mutation or mistakes. And so very few things are entering and leaving the nucleus. And so when we're talking about the nucleus, we also need to talk about the structure of DNA, specifically in human cells. So you've probably heard of the idea of chromosomes, or you definitely have. You've all taken Bio 160 or some introductory bio course. So chromosomes in humans exist in pairs. We have 23 pairs of chromosomes, so 46 chromosomes total. And a chromosome is essentially a highly structured and condensed set of DNA that codes for a known set of genes. So in every chromosome pair, you have two separate chromosomes. One came from the father, one came from the mother. Each of those chromosomes carries DNA that codes for the same genes. So you would find a gene for hair color on both of the chromosomes in each pair. However, the DNA in each individual chromosome for that gene could be different, right? You could have, and we call those alleles. So you could have the allele for blonde hair on the chromosome that you got from your mother, and you could have the allele for brown hair on the chromosome that you got from your father. And then the interaction of those alleles ends up turning into the phenotype, such as my brown hair, right? I could either have two copies of that gene, or I could just have a single allele of that gene because brown color is dominant as a gene. So anyways, chromosomes themselves are these highly condensed structures of DNA, and they exist in pairs. So each pair has two different chromosomes that carry the same genes, but they could have different alleles for those genes. If we further unravel these chromosomes, the centromeres and kinetochores are only present during cellular replication, and this fully supercoiled chromosome is really only present during mitosis, during cellular replication. But if we unravel this supercoiled region of the chromosome, you can start to see the proteins that are holding the DNA into this tightly coiled shape. And so those proteins are called histones. They're very circular little proteins. And we just use those to wrap our DNA around it. And so 8 histone proteins with their DNA attached is referred to as a nucleosome. So DNA wraps around histones. Histones come together to form these nucleosomes. Multiple nucleosomes end up coiled tightly together into chromatin, which can be further coiled into a chromosome. So you see that here. Like I said, what's being depicted here is during cellular replication, where all of your chromosomes have been duplicated. And so when you have an exact copy or a clone, a duplicated version of that chromosome, that is referred to as a sister chromatid. And they attach to each other through these kinetochores and their centromere. And so each kinetochore is where a spindle fiber will attach and then pull those sister chromatids apart. And we are going to talk a bit about mitosis here. So having that understanding is important. So there's a difference between homologous or between chromosome pairs versus sister chromatids. Chromosome pairs code for the same genes, but could have different alleles. You have 46 individual chromosomes with 23 pairs. During replication, every single chromosome, so all 46 chromosomes, are being duplicated into sister chromatids. So we would have 92 sister chromatids present while preparing for replication. And so these are clones of each other, right? And they would be connected at a centromere through these kinetochores. And so during mitosis, all of these are going to line up on the metaphase plate. I guess because I'm skipping ahead, let's kind of get to cellular replication. Sorry, I didn't realize how far I skipped ahead here. So after all the chromosomes have been duplicated into their sister chromatids, those sister chromatids will line up on the metaphase plate. See here? So they kind of line up in the center of the cell. And a bunch of spindle fibers, these microtubules, are going to extend from their centrioles and attach to each kinetochore. And then they will start to shorten, which pulls each sister chromatid apart so that you get one copy of those original chromosomes going to each side of the cell. And so you'll end up with the same 46 chromosomes that you started with in each of our new cells. And so you've now made clones of the original cell by duplicating all of that DNA. So one sister chromatid goes in each direction. So you end up with 46 chromosomes with their same 23 pairs on each of the daughter cells that were created. And that's basically mitosis, and that's essentially all we're really talking about with cell division anyways. Okay? The goal of cell division is to create new cells. So you need to duplicate all the DNA and then separate that DNA into two sides of the cell. And then that cell, that large cell with all this DNA is going to pinch off until you get two clones of the original. And that's mitosis in a nutshell. And that's enough for what we need to discuss related to that. So the other process that I skipped over here related to our nucleus is the role it plays in protein synthesis. So like I said, your genetic material, all of your DNA, all of your genes, they are simply coding, they are instructions that code for the synthesis of proteins, the building of proteins. So all of the genes you inherited from your parents simply describe what proteins your cells are going to make, and that leads to the functions that your body is capable of, as well as all of the physical characteristics that we associate with our genes, right? Hair color, eye color, etc., skin color, height, some things related to weight. That is all based on the proteins that your DNA codes for. So what is considered the central dogma of biology, which describes the basis of how living things function, is with these steps. So all of our DNA, the genetic material of living things, is going to be transcribed into a copy of a gene. So whatever segment is going to code for that protein is going to be transcribed into mRNA or messenger RNA. That messenger RNA will take that short transcript to a ribosome, and the ribosome will translate those instructions into a string of amino acids, which will eventually become our proteins. And that protein then carries out that gene. So I'm not going to ask you too many things related to the specifics of transcription and translation, such as recognizing RNA polymerase and the directionality of DNA and all of that. You probably learned about that in 160, and you'll get other practice with that if you're going more specifically into genetics and evolution stuff. But for the purposes of this course, just generally recognizing how these processes work is important. Okay, so how your genes lead to a functional protein. So your DNA, the segment of that DNA that codes for a gene, is going to be unraveled from its histones, and then the two complementary strands are going to be pulled apart. And that is going to allow for an mRNA transcript to be created from that template strand. So by the end of transcription, we have made a copy of the short string of DNA that codes for a single gene or a single protein. That mRNA transcript will then leave the nucleus and go find a ribosome. Go find that. Sorry, I said that strangely. It will go find a ribosome. There you go. That could be a free-floating ribosome in the cytosol, or it could be a ribosome that is directly attached to the rough endoplasmic reticulum. And that ribosome is going to translate these instructions using every codon to add one amino acid. So codons are groups of 3 nucleotide chains, right? So AUG is considered our start codon. Right? So these are always nucleotide triplets, 3 nucleotides for every 1 codon. And each codon is going to match with an anticodon that is on tRNA. So messenger RNA is carrying the transcript. tRNA, aka transfer RNA, is going to bring the relevant amino acid that matches that codon. So the ribosome is going to help with matching up these codons from the mRNA transcript with the anticodon carried by a tRNA, and that tRNA is bringing the amino acid that these codons code for. And so this process is essentially going to repeat. The mRNA transcript is going to continuously move down, and a tRNA is going to come match with it and add a new amino acid to our growing chain. So they will form a peptide bond. And now that empty tRNA will leave, and a new tRNA molecule will come read the next codon. It'll add our next amino acid. There will be a new peptide bond formed between them, and this will repeat until the transcript is done or it reaches the stop codon. And so by the end of this process, we will have a completed polypeptide in its primary structure, a simple string of amino acids, and that is the beginning of a protein coming to life. Right? So this string of amino acids is just the primary structure. They will start forming hydrogen bonds to create that secondary structure, and then the endoplasmic reticulum or the Golgi apparatus will help create the disulfide bonds that give it its 3-dimensional shape, its tertiary structure, and or add multiple polypeptides together into their quaternary structure until you have a functional protein. And that is the basis of how all living things work. DNA transcribed into mRNA, mRNA translated into a string of amino acids. We're not going to do practice with that. I'm not going to ask you to use a template strand of DNA, transcribe that into the template mRNA, and then translate that mRNA into a string of amino acids. But that is a useful skill to have, especially if you are interested in learning more about genetics and evolution. And so I just have that reference there. So that should wrap up all the major topics that we needed to review from chapter 3. Just like with the last chapter and essentially with all the chapters in this course, I'm not discussing every single thing mentioned in the chapter of the textbook, so it is still useful to read through that, but I should have covered all the major topics that you would see on an assessment, on an exam. So please work through this stuff, review some of the slides, after you've watched this video, go through the quizzes, look through the additional resources that I've offered to you. And if you still have questions, or you feel like you don't know how to properly address any of the learning objectives or the things from the study guide, please, please, please reach out to me so that I can help. And yeah, other than that, I will just see you guys in the next video.